guys. So we are going to be working on part 2 of our notes for this week's homework. This is another fairly short set of notes. We are going to be looking specifically at DNA replication and how that all happened and how we figured out what DNA looks like, and we'll be working on eventually making some of our own DNA models. So hopefully you see your DNA replication worksheet in the Google Classroom. And let's go ahead and get started. So we start here. Let's present. Again, print it out, write on it, however you want to do it is fine with me. But just make sure you turn it in. Do not copy any of this part down. This is more for background information only. So DNA has a very interesting history. Back in 1928, scientist named Griffith was looking at the transformation of one bacteria into another. Kind of got the idea that there was something going on. There was some way that information was being passed from one thing to another. Then in 1944, we have the idea that DNA is the molecule responsible for those transformations that they had seen, and that it makes up the genetic material in cells. Then we get to a really big breakthrough, and that was in 1952, Hershey and Chase. These two scientists confirmed that DNA is that molecule that makes up the genetic material of the cells, and we will talk about Hershey and Chase on our next slide. And then we have Chargaff's rules. So Chargaff took a look at everything and figured out that in all species, the DNA goes in certain proportions, and we have adenine as a nitrogenous base. There are equal amounts of adenine to thymine, and there are equal amounts of cytosine to guanine. And those are abbreviated by their first letters. So adenine is A, thymine is T, and cytosine is C, and guanine is G. In the 1950s, there was another scientist named Rosalind Franklin, and we're going to watch a whole video on her. And she used X-rays to photograph the very first photo of DNA. In fact, she didn't even realize she had done it until work was published later on that by Watson and Crick, there was this race to see who could figure out what DNA looked like first. And they published information and they built their model based on seeing one of her pictures that she hadn't even looked at yet. And very interesting story between all of that. There's a very good movie we will watch on that. I really like to spend a little bit of time giving Rosalind some credit for what she found. So her discovery predated Watson and Crick's discovery, and Watson and Crick wouldn't have been able to do what they did in the time that they did without her information. And we'll get more on that in a few minutes. All right, so here's the Hershey and Chase experiment. These guys were trying to discover or determine where the genetic information of of a cell is stored. They wanted to look at the outside, at the protein structure, and the inside, which they thought was the DNA, which was where they thought the genetic information was stored. So they looked at bacteriophages, which are a type of virus that can infect bacteria. They were able to mark part of the virus with a P, so phosphorus, so they made it radioactive, and then part of it with the S, which is radioactive sulfur. They mixed them up, did a whole experiment. We're going to watch a video on it in just a second to find out which part gets put from the bacteriophage into the bacterium. Okay, so let's watch this real quick.
In 1953, Alfred Hershey and Martha Chase confirmed DNA's preeminent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
Subscribe to my YouTube channel, The Bio.
Okay, so that gives you a little bit background of that particular experiment, and that's why it was really fundamental that we understand what is getting transferred so that we can figure out that the structure and know how the genetic information is going from one thing to another. So it's a very important discovery that is actually kind of hard to understand if you just looked at this picture. So that's why I showed you the quick video of it. Nope, you don't want that to play. No. There we go. All right, slide 4, the role of DNA. So DNA has 3 roles. You need to make sure you know all 3 roles. The first is to store information, the second is to copy information, and the third is to transmit information. So storing it, DNA stores information for genes that control patterns of development, from eye color to hair texture. All of those things are stored in your DNA. To copy, we know that this happens in the cell cycle during the S phase. You have to make a complete copy of every single one of those genes, and then those checkpoints are also there to make sure that the DNA has copied correctly and is the correct length as well. And then information from one cell goes to multiple cells, either through replication or through like the bacteriophage injecting it into another cell. So that's how we get genetic information transmitted. Okay. Nucleic acids and nucleotides. This is in your book in chapter 2.3, but we're not going to worry about that necessarily because we didn't do chapter 2. Nucleic acids are long molecules that are found inside your nucleus. A nucleic acid has nucleotides as part of it. Nucleotides are made up of your nitrogen base, your deoxyribose sugar, and the phosphate group. So we have this structure here, and you have your nitrogenous base here. There's A, G, C, and T, adenine, guanine, cytosine, and thymine, connected to the sugar here, and then the phosphate here. This makes up your sugar-phosphate backbone. Your sugar connects to the phosphate of the next, the phosphates connect the individual ones together. So this phosphate connects both of those groups. This phosphate connects both of those. All right, 4 nitrogenous bases. Your A's are your adenines, your guanines are your G's, your cytosines are C's, and your thymine is your T. Make sure you have this is down as your backbone. So on your notes, there's that arrow, the bracket, add in backbone. Okay. Chargaff's rule. Super important. His rule says that the percentage of A equals the percentage of T, and the percentage of G equals the percentage of C. And that's because they can only partner with each other. So if you can only have A's going with T's, if there's 30% A's, then there has to also be 30% of T's. So hopefully that makes some sense, which means if you have 30% A's and 30% T's, 30 plus 30 is going to be 60%, and that means you have 40% for both G and C. So then divide that by 2 to get 20 and 20%. And we'll talk about that in a little bit. Actually, we'll probably do that when we get to our guided practice. Okay. All right, here's some Rosalind Franklin information. She was a British scientist who found this X-ray and was in her, so she had an A pile and a B pile basically. That's the gist of it. And her B pile was her not really important, not relevant pile. She actually had this photo in there, did not realize what she had discovered, and had basically not discarded, but she was not sure. She believed that her photo, the photo that she wanted, was going to be in this other pile. And then, you know, the rest is history, and we'll watch a video on that one. And this is what the pile, or the photo, looks like. And you can see this X means that there's a helix shape because it's looped around. So you have, you can see the lines at a given moment like that forming. Okay. All right, Watson and Crick. I am not a fan of what they did, if you have not gathered already. So they stole her photo, and they didn't like physically steal it. They just sketched it, and I believe it was, I think it was Crick. Now I can't remember. One of them did it. Only one of them went over to her office. One of them copied it down and brought it to the other person. It's in the video again. It's just been a couple years since I've seen it, so I just know it was one of them, and I always lump them together, and that's probably not correct. They were spending a lot of time trying to come up with a 3D model of DNA. They couldn't make it work. And it wasn't until they were able to get their hands on this image that they discovered that the base pairs were actually on the inside facing each other. They kept doing models with them on the outside, and they just couldn't get it to work. So once they flipped them and got them on the inside, they were able to correctly model what DNA looks like. And then, not only did they correctly do it, they published it, they got Nobel Peace Prize for it. They did not give Rosalind credit. So that was definitely something that was scandalous. Now that we all know what was going on, a lot of us didn't know for a very long time. So it's definitely something that is scandalous, so to speak. Okay. All right, hydrogen bonding. This is important. This is how our two halves of a DNA strand come together, that bond between the base pairs. You will notice there are 3 bonds between the G and the C, only 2 between the A and the T. And that's how you can tell. There's going to be a worksheet that we will work on, and it gives you just the lines, and that will help you figure out which molecules go where. So 3 is G and C, 2 is A to T, okay? We're not doing the DNA models, so this is instructions for in class if we were going to be doing that, okay? So also, one more thing to note, hydrogen bonds are represented by a dashed line. It is not a super strong bond, certainly not as strong as like a covalent bond or an ionic bond, and those are ones we will be you will talk about when you take chemistry, okay? So Watson and Crick realized that base pairing explained Chargaff's rule, and it gave a reason for why A and T percentages or concentrations, these brackets mean concentrations, equal each other. So they were able to solve the structure of DNA by stealing Rosalind's photo and information, and then using Chargaff's rule to help explain everything. Okay. And DNA replication. So you have a copy of this picture on your notes packet. Replication is going to occur during the S phase of the cell cycle. DNA is duplicated before cell division occurs. That's really important. You can't have a cell dividing if the DNA isn't already duplicated. So we hit that hard when we did mitosis and we did cell cycle. Mitosis is after the replication of DNA. So make sure you know that that is occurring in the S phase. Okay. When you have your DNA strand, if you've got 2 strands like this, it opens up at a replication fork, which is right here. Each original strand is now called the parent strand, and they both serve as a template for a new strand to go on. So you have one strand here turning into two strands here. Okay, does that hopefully make sense? If you are having trouble understanding this, please come talk to me during pause or in class. It would be a great opportunity to talk about it. It's not easy to understand necessarily. And there's an enzyme on here called DNA polymerase, and that is the one on here that is going to be responsible for a couple of things, proofreading and some other things. We'll talk about that in a couple of slides as well. Okay? Next, both strands of the double helix separate or unzip. They form a replication fork. In eukaryotic cells, this is going to occur at multiple locations on the same strand of DNA. New bases are added following the base pairing rules, and yeah, you'll have new strands being replicated all over the cell. Each DNA molecule resulting from the replication has one of the original strands and one new strand. So that hopefully is making some sense. We've kind of hit this a few different times now. So we have our original strand on the outside, new strand on the inside, the replication fork right here, meaning it's not yet paired. Okay. Now, there's a couple of enzymes that are involved in this. The only one we're going to focus on today is RNA or DNA polymerase. The other one that we that there is involved with this is helicase, and that's the one that unzips and causes the replication fork to form. But DNA polymerase is the one that joins the nucleotides to produce that new strand. So it sits right there where there's an unpaired one, and it kind of allows the other one to attach. It also proofreads to make sure that that's happening correctly. So this part is going on in the G2 phase, the proofreading part, right? It's making sure that there's no errors, okay? Because that's the checkpoint for number 2, or G2 checkpoint is to proofread. Okay, telomerase. This is another enzyme, or not enzyme, sorry. This is the end of the chromosomes, is called telomeres. Telomerase is an enzyme. It adds short repeated DNA sequence to the telomeres to lengthen those chromosomes, and that's because as they replicate over and over again, little bits of it are lost, so it gets shorter. So you have to keep adding to the telomeres. So I can't talk. Telomerase does that, and it reduces the likelihood that gene sequences will be lost during replication. So it keeps this buffer on at the end of the chromosomes, okay? So, replication of living cells. Replication always occurs before cell division. Hopefully we've got that. It occurs in the S phase. Mitosis is when, in the M phase, when the cell divides. Prokaryotes do it a little differently than the way we talked about with eukaryotes. Prokaryotes have that single circular strand, right? And they have it just free floating in the cytoplasm. It's not enclosed within a nucleus. The replication starts from a single point, but it goes in both directions. So you only have replication occurring from one spot going around. So it's not going to be doing multiple places where eukaryotes are, and they can do it up to 1,000 places, okay? Or I'm sorry, they have up to 1,000 times more DNA, so they need to be able to do it more quickly, so they have lots of places for it to all do replication. It just makes it hundreds of places, makes it faster and more efficient, okay? So lots of replication forks going on there. I think that covers everything in here. I tried to keep it as short as I could. Sorry, it's still 19 minutes. Have a great rest of your day. Make sure you turn this in, okay?